in vitro production, analysis, characterization of betalains from Talinum triangular jacq wine, a valuable medicinal herb. So this was a part of my PhD research work which I carried out in the Department of Plant Biology and Biotechnology, Loyola College. So some basic introduction. Plants, they produce a wide array of colorants, and these natural pigments can be classified into two different categories, the water soluble and the fat soluble pigments. The commonly known anthocyanins, the betalains and the carminic acid can be classified under water soluble pigments, while the carotenoids and chlorophylls are classified under the fat soluble pigments. So the term betalains was first coined by Mabry and Reading in the year 1968 after they were first isolated from beta vulgaris, that is the common beets. These indole-derived plant pigments are also known as chromoalkaloids, and they get mainly accumulated in certain plant parts such as the flower, stem, leaf, root as well as in the fruits. So this betalain pigments are characteristic to the plants belonging to the order Caryophyllales, and except for the two families, that is Caryophyllaceae and Molaginaceae, which accumulate anthocyanins instead. They are also found in certain higher basidiomycete fungi such as Amanita and Hygrozybe. So, a little of chemistry of the betalains. So, the chromophore of the betalain has been described as a protonated 1,2,4,7,7 pentasubstituted 1,7-diazaheptamethane system. So, this is the basic structure of the betalains, and the part below the dotted line represents the beta-lamic acid moiety. So, based on this, it has been classified into two categories, that is the red-violet beta-cyanins and the yellow-orange beta-xanthins. So, these beta-cyanins are nothing but they are the ammonium conjugates of this beta-lamic acid with cyclodopa, while the beta-xanthins are the conjugation between this beta-lamic acid with either amines or amino acids. So there are a total of, so basically the betalains can be identified as a beta-cyanin or a beta-xanthin depending on the identity of the R1 and the R2 residues. So a total of 50 beta-cyanins and 20 beta-xanthins have been found to occur naturally, and all of them have the same basic structure. The difference among them are attributed to the presence of different sugar or acyl groups for beta-cyanins and the presence of different amines and amino acids in beta-xanthins. So this wide variation in color is mainly attributed to the conjugation of this substituted aromatic nucleus to the chromophore. This shifts the absorption maxima from 480 nanometers, which is the yellow color for the beta-xanthins, to 540 nanometers, which is the pink color for the beta-cyanins. So certain functions of the betalains, some of them are they help in attracting pollinators, they help in promoting seed dispersal, they prevent damage that is caused by pathogen infiltration and wounding. Mainly these betalains are used for coloring low acid foods as they are water soluble, and also they are stable over a wide pH range of pH 3 to pH 7. They are also known to have remarkable antioxidant capabilities, antifungal activity, anti-inflammatory and anti-carcinogenic activity. So why use these betalains? So these natural colorants are gaining more interest because artificial colorants are becoming more critically assessed. So these nitrogen containing pigments such as betalains are used as non-toxic food colorants, and they are also used in other industries such as cosmetics, pharmaceutical industries, etc. So betalains have also been proven to have higher bioavailability when compared to flavonoids, as well as superior stability when compared to anthocyanins. As we all know that the major source for betalains are beta vulgaris. However, they have certain drawbacks. Some of them include limited pigment spectrum, an earthy smell due to the presence of the odorant geosmin, adverse flavors which are caused by various pyrazines, high nitrate levels which can be carcinogenic when it is ingested, also the carryover of soil microbes leading to microbial contamination. So because of this, we are looking into alternative betalain containing sources. So some of them include the Amaranthus Gomphrena globosa, Celosia, all of them belonging to Amaranthaceae, the Opuntia hylocereus from the Cactaceae family, beta vulgaris subspecies Cycla from Chenopodiaceae, the Bougainvillea from Nyctaginaceae, Mesenbryanthemum from Isoaceae, and Portulaca grandiflora, which is the Portulacaceae member. So all of them, they have their own advantages and disadvantages because some of them are edible while the others are not. So based on this, this plant, that is Talinum triangula, which is a Potlaceae member, was selected based on the availability of the plant, the soil and the climatic conditions under which they are able to grow, as well as the ethnobotanical information for the treatment of various diseases. So this is just the morphological features of the plant Talinum triangular, and this is the scientific classification, and some of the synonyms for Talinum triangular are Talinum fruticosum and Potlaca fruticosa. So a little about the plant description. The Talinum triangular is a short-lived erect perennial herb belonging to the family Potlaceae. It is popularly known as water leaf because of its high moisture content of almost 90.8% per 100 grams of edible leaf. So the purslane family is a group of edible plants with cosmopolitan distribution, and the genus Talinum comprises of 15 species, of which only 5 species have been reported in India. The growth habit, it's a shade-loving plant, and it is characterized by thick succulent stems with pink-colored flowers borne on a triangular peduncle. Hence, it gets the name Talinum triangula. The origin, it is known to have originated in Africa, but is now cultivated throughout the humid tropical countries. However, it was first introduced into Tamil Nadu from Sri Lanka. Hence, it gets the vernacular name Ceylon spinach. The ecology, it is a drought-resistant plant, and it adopts a crassulacean acid metabolism pathway. And the uses, it can be either cooked or it can be eaten raw. It can be made into soups, salads, stews, etc. So these are some of the health and medical benefits of tea triangula. It has been implicated in the management of cardiovascular diseases like stroke and hypertension. It is also used in the treatment of diabetes, jaundice, obesity, etc. So some of the objectives of the present study are to produce and to characterize the betalains from both in vivo as well as the in vitro samples of tea triangula, to quantify the betalains by using a UV visible spectrophotometer, to investigate the beta-cyanin and the beta-xanthin patterns by HPLC-MS analysis, to assess the betalains by using in vitro biological activity, and finally to determine the storage and the viability of the pigment. So this is just the basic outline. So the research work is divided into two categories. One is the in vitro production, the other is the in vivo production. Under in vitro, two methods were adopted. One is the callus culture, other is the liquid culture. For in vivo production is the naturally betalains being produced in the plant, especially in the stem, flower and the leaf. Once the betalains were produced, they were then extracted. The method of extraction was standardized, and after the betalains were obtained, they were then subjected to quantification. For this quantification, we use a multiple component method, and this method was also standardized, and they were expressed in terms of the beta-cyanins, the beta-xanthins and the total beta-lane content. So for the analysis of the beta-cyanin and the beta-xanthin patterns, we used a reverse phase HPLC-MS analysis, and further the in vitro beta-lane studies were carried out. So for this, the antimicrobial activity, thin layer chromatography, bioautography, cytotoxicity using a hemolytic assay method, and the in vitro antioxidant activity was calculated. So finally, the applications, the storage and the viability also was determined. So the presentation has been divided into two parts. So the first part is the production and the characterization of the betalains, and the next part is the in vitro studies, and finally is the applications and storage. So this is the first part. So here, as you can see, the betalains which are produced naturally in the flower, the stem as well as in the leaf. This just shows the localization of the pigment in the stem on a cross section, and it was seen only in the epidermal cells. So for all the studies, the epidermal peel of the stem alone was used. So these are just certain qualitative tests that were carried out, where it is just to confirm that betalains were the major pigment, and the addition of NaOH and HCl turned the color to either yellow or it destroyed the pink color. So this is the quantification results. So 4 different buffers were standardized, and here it was observed that 80% methanol was the best extraction buffer for extracting maximum total betalain content of almost 2.28 mg per gram fresh weight. So this is the quantification results for the in vivo samples, that is the flower, stem and leaf. So here both fresh samples as well as dried samples were tested, and the results showed that the dried floral extracts yielded maximum betalain content of almost 10.7 mg per gram dry weight. Moving on to the in vitro production. So here, as I said before, it is the two different techniques were adopted. One is the callus culture, the other is the liquid culture. For callus culture, different explants such as leaf, internode and transverse thin cell layers were used, and they were subjected to surface sterilization, and they were inoculated onto Murashige-Skog MS medium containing different concentrations as well as combinations of plant growth regulators. So here the parameters that were studied were one cytokinin with either one auxin or a cytokinin with two different auxins. For the liquid culture, the explants that were used were leaf, shoot tip and node, and here also after surface sterilization they were inoculated onto liquid MS medium. So here three different parameters were studied. One is the cytokinin auxin combination, cytokinin in combination with another cytokinin, as well as the MS salt concentration, that is both half strength MS and full strength MS. So this plate shows the production of the betalain from callus culture. So we can see both the variation in the callus morphology as well as in the color that is being produced. Both friable as well as compact callus was obtained. So this is just a magnified view of the same callus as observed under a dissection microscope. This is the quantification results, and this shows the MS medium containing BAP in combination with NA and IBA yielded maximum total betalain content. This is for the betalain production in liquid culture, and this showed the elicitation of the betalains into the medium. So this is the quantification results, and here it was seen that full strength MS having BAP and NA as the hormonal combination yielded a total betalain content of almost 4 mg per ml. So next is the spectrophotometric analysis. So a full wavelength scan of all the samples were carried out, and it was noted that a major peak was observed at 540 nanometers. So this peak corresponded to the lambda max, that is absorption maxima of beta cyanins, the beta nin. So this was further confirmed by a HPLC elution, and this is for the in vivo samples, the flower, stem and the leaf. Both were monitored at 480 as well as 540 nanometers. So two major peaks were seen, one at the 20th minute and the other at the 21st minute. And however, this was absent in the callus culture but present in the liquid culture. So these are the mass spectrometric data. So this revealed that the 20th minute peak corresponded to the beta cyanin beta nin having a lambda max of 538 nanometers and a mass by charge ratio of 551. The 21st minute peak corresponded to the isomer of beta nin, that is isobeta nin, having a similar mass by charge ratio. These are the mass spectrometric data for the in vitro samples. And this is just the positive ion spray mass spectra for the beta nin and isobeta nin, both having a parent ion scan at 551 and a daughter ion scan at 389. So next part is the in vitro biological activity. So first, for the antimicrobial activity, 5 gram-positive bacteria, 7 gram-negative bacteria and 6 fungal pathogens were tested against these betalain extract, and this was carried out by both distribution method and MIC. So among them, the betalain extract showed maximum inhibitory activity against these 4 bacterial pathogens alone, and significant activity was observed against E. coli having a zone of inhibition of 17 mm and an MIC value of less than 0.078 mg per ml. So these are the TLC and biotography results, where 14 different compounds were separated on the TLC plate, and this is the biotography results showing clearing zones at various RF values against these 4 pathogens. So this is the cytotoxic assay. This was done by using a hemolytic assay method, that is against human RBC cells. And here 50% lysis of the RBC cells was observed at a concentration of 38.4 mg per ml, proving that the betalains were non-toxic even at very high concentrations. Next is the antioxidant activity. So here both non-enzymatic as well as in vitro free radicals were studied, and so some of them are the polyphenols, phenolic acid, flavonoids, flavonols, and the free radicals include the total antioxidant capacity, reducing power, TPPH, ABTS, nitric oxide, hydroxyl radicals, superoxide, and metal chelation. So this graph shows the results of the non-enzymatic antioxidants, where both the polyphenol and the phenolic acid content in the betalain was calculated and expressed in terms of gallic acid equivalents, while the flavonoids and flavonols were expressed in terms of quercetin equivalents. So this is the results for the total antioxidant capacity and reducing power. So here there was a concentration dependent gradient for both the extract as well as for the positive control. So this is just the free radical scavenging assay, and here it was observed that betalains were able to scavenge nitric oxide radicals, and the results were almost equal to that of the positive control ascorbic acid. So finally, the applications, the storage and the viability. So the betalain extract was extracted in water and then added to certain food products such as milk, ice cream, and commonly made South Indian sweet known as kesari. So here basically there was only enhancement in the color of the final food product. However, there was no change in the flavor or the taste of the food product. So for the storage and viability, different conditions were tested. Some of them were the light and dark conditions, as well as room temperature 4 degrees and at minus 20 degrees centigrade. And here it was observed that the extract, when it was stored at minus 20 and at dark conditions, were viable even after a period of 3 months. However, the best method of storage was as dried extracts and can be stored for more than a year and freshly extracted before use. So the summary. The betalain profiling of the in vivo and the in vitro samples of T. triangulae for the estimation and detection of beta-cyanins and the beta-xanthins was attempted for the first time. The photometric quantification and the HPLC-MS analysis was able to determine that the beta-cyanin patterns in flower, stem, leaf and the liquid culture was dominated by betanin and its C15 diastereomer, the isobetanin, as the principal red pigment. So the various in vitro assays also corroborated that the betalain extracts possessed potent antimicrobial and antioxidant capabilities. Altogether, the findings of our study may open up new opportunities for screening of betalain containing plants such as T. triangulae floral extracts as promising future sources of natural food colors. So the conclusion and future prospects. So medicinal plants are traditionally a part of our human diet, containing naturally occurring compounds that may exert physiological effects beyond nutrition by promoting human health. So the main focus of this research work was to achieve maximum benefits from a universally available edible plant. So the betalain profiling of Talinum triangulae was carried out to enlighten common man with the multifunctional aspects of this plant. The potent antimicrobial as well as antioxidant capacity further augments the implications of this study as a natural food colorant. Therefore, the current work could be considered beneficial for the sustainable utilization of Talinum triangulae and its applications in pharmaceutical and food industries. So these are some of the references which have been cited in this presentation, and some of my other publications. Thank you.